Hi, thanks for the introduction. Thanks to the organizers for letting me speak in this session. I talked a little bit about quantifying fusion last year at the conference, so I decided to shift my talk a little bit for today's session. I hope anybody reading the abstract doesn't get too disappointed. So my studies for my graduate work are focused on HAP2-mediated cellular fusion in Tetrahymena thermophila. So membrane fusion is a process in which separate lipid bilayers merge to create a single continuous membrane. And usually this occurs under the direction of specific proteins through the formation of a hemifusion intermediate. And among all of the membrane fusion events within an organism's lifetime, I would argue that perhaps the most defining, crucial, and exquisitely controlled is that of gamete membrane fusion, which occurs during sex. And sex is important. It doesn't just define us as humans, but it defines us as eukaryotes. But unfortunately, there's not a lot known about the molecular interactions of gametes and especially the fusion proteins involved in this process. So in order to study these types of events, you need a good model system that allows access to a lot of gamete material. And we work with the Ciliate Tetrahymena thermophila, which, although it doesn't form true gametes, can be sexually differentiated and easily genetically manipulated and mated in mass cultures. And sex in Tetrahymena occurs through an adhesion of complementary mating types under starvation conditions, and is unique compared to those sexual events in other organisms in that these cells will form a specialized region of membrane called the conjugation junction, in which not one but hundreds of fusion pores are formed. And they're non-expanding fusion pores that, instead of fusing the two cells completely together, are resolved at the end of mating such that the two cells can separate into individually maturing ex-conjugate cells. So to study this process, we, in collaboration with several others, and most importantly Dr. Eric Cole, who is in the audience and will be giving the talk directly after mine, decided to look at this highly conserved, putative, ancient gamete fusion protein called HAP2 in Tetrahymena and examine its function. Now, HAP2 has orthologs in many other species, and it's widely diverse present in different phyla, from single-celled protists all the way up to invertebrate animals and flowering plants. And we found in that first study 2 years ago that HAP2 localizes to the conjugation junction between mating cells and is expressed and functional on both cells of the mating pair. However, you need to knock out HAP2 in both cells of the mating pair in order to result in a complete block to progeny formation and fusion pore formation. And this is unlike in other species in which HAP2 has been studied so far, because in other species it's only present and active on the male gamete. So it's present on both cells of the Tetrahymena pair and functional. But after this, progress was kind of difficult because relatively little is known about the domain structure of this protein so far. And we wanted to look at which domains might be important in regulating HAP2 function in fusion in Tetrahymena. And so we decided to do predicted structural modeling using this online server FIRE2, and found a striking 95% confidence homology to the dengue virus envelope glycoprotein. And this is a class 2 viral fusion protein. And this prediction covered a wide swath of the ectodomain of Tetrahymena HAP2, about 166 residues. And I repeated this type of template-based modeling on other platforms that are available online, CPH Models 3.2 and RaptorX, and found that they all repeatedly found the same hit to a class 2 viral fusion protein structure. And the hits were high enough that you could build predicted tertiary structures of the Tetrahymena HAP2 protein. And these predictions are of the ectodomain and are colored in accordance to the domain structures of typical class 2 viral fusogens. And you can see on the side that the dengue virus envelope protein, actually, you can see the resemblance here. And before I move on, I want to highlight this region at the very tip of domain 2, which is called the fusion loop in the viral fusion protein. You can see it's picked up in some of the other predictions here and here. And this is a very critical part of the viral fusion protein that allows basically punching into the target bilayer that's going to be fused to initiate a lipid mixing event between, and is really the action zone of this protein. So now we had some putative domains to go after in our truncation studies, but we needed a good assay, and it's easy to make these types of truncation mutants in Tetrahymena, and I would place them back into the endogenous locus so they're under control of the HAP2 promoter, but we didn't have a good assay. And I won't delay on this too long, but we found that you can label the cells with fluorescent cell tracers and see exchange between different mating partners after mixing of these fluorescent cell tracers, and that allows, after the X conjugates separate, to look for the presence of these markers in crosses. And wild-type crosses, you always see exchange. Knockout by knockout crosses, you don't see exchange of these tracers. And you can quantify this using flow cytometry, and you can see this gated population here. About 80% of cells have experienced fusion, and while its knockout knockout pairs do not. So we looked at these various truncations and knocked out the HAP2 domain, and then the FIRE2 predicted entire region of homology to the dengue virus protein, and didn't see, saw a lack of fusion in these mutants, but these are fairly broad truncations, and we believe they affected the protein folding potentially. So we did immunofluorescence on all of these mutants to look for localization. And if you knock out the fusion loop, though, importantly, you don't see any fusion in these mating cells. But replacing the HAP2 fusion loop with the dengue viral fusion loop also doesn't rescue. So there's a difference between these fusion loops structures. And then we made some targeted mutations around the fusion loop in different amino acids that were conserved, and probably didn't hit the right ones because these all could fuse. But these cysteines here are predicted from, at least our predicted structures, to be part of a disulfide bond stabilizing the loop, and those showed a complete lack of fusion as well in our truncations. So we've tested the functional extent of this homology, but we were looking for an additional way to look at how the HAP2 fusion loop might be interacting with membranes. And so we teamed up with Dr. Alex Lai and Dr. Jack Fried, who Dr. Jack Fried runs the ACERT lab at Cornell in chemical biology, and they do these really interesting, awesome studies with this technique called Electron Spin Resonance to measure the interaction of viral fusion peptides with artificial membrane bilayers. And so we synthesized the HAP2 fusion loop peptide along with controls being the wild type dengue viral fusion loop, influenza fusion loop wild type, and then mutants of these viral peptides that are known not to interact in these assays with the membranes or in functional studies. And these mutants of the viral peptides are circled here. So this assay relies on making an artificial membrane that is composed of and has in its composition these spin-labeled lipids, which have nitroxide groups either on the phospholipid head or within the acyl chain, that the detector can sense the changes in the membrane composition based on where these spin-labeled groups are positioned. And when you have an interaction of a viral fusion peptide with the membrane, they've repeatedly seen that there's a slight dehydration in the membrane due to increased phospholipid packing, So you get an increase in the order parameter of those membranes when you add in increasing concentrations of peptide. And surprisingly, we actually saw the HAP2 fusion peptide increases the order parameters of these artificial membranes up higher than the dengue viral fusion loop peptide does in both membranes composed with phospholipid head spin label and those with an acyl chain spin label, while the mutant peptides do not increase the same level of order parameter. Next, we did an assay that relies on the relief of a self-quenching R18 lipid in a bulk fusion assay. So when the quenched liposomes are fused in the context of peptide with an unlabeled liposome, you get a fluorescent readout. And we saw that wild-type fusion peptides as well as HAP2 induce this type of lipid mixing and increased fluorescence, but in mutant conditions, they do not increase the same level of fluorescence dequenching. So we found that there's a functional and structural link between HAP2, the HAP2 fusion loop, in also basically that this HAP2 protein has a link to these viral fusion proteins. And the question now is what is this potential link telling us about eukaryotic sex? And so I just want to muse at the very end here that so while we know that the sex is maintained in populations by endowing species with an enhanced ability to basically adapt to changing environmental conditions through increased spread of beneficial mutations throughout the population, this mode of maintenance is through group selection, which doesn't explain, and it makes it very difficult to understand how sex could have been initiated originally in the first cells ever to try it. So one theory by Donald Hickey in 1982 suggested that parasitic genetic elements such as transposons or viruses could promote the initial sexual cell fusion events in order to enhance their own transmission through early eukaryotic populations. And I think with this connection to viral fusion proteins, we might want to reconsider this theory, although it's very difficult to test. So thanks to everyone in my lab, especially Ted, Donna, Daniel, Kelsey, and Gary, and all of our HAP2 collaborators, past and present, Dr. Eric Cole, everyone at UC Boulder, and especially for this current work, Dr. Alex Lai and Jack Fried at Cornell, and my committee members and my funding sources for support. Thank you.
I see that you tested the fusion peptide loop or fusion peptide is essential and sufficient to make the bilayer membrane to fuse. Have you tried to use it to force the two knockout cell to fuse together, or you finish conjugation?
I haven't tried that yet. That's a good idea. I don't know if it would work.
Essentially, what's the rest of the protein?
So I've been trying actually to inhibit fusion with some soluble proteins, and the cells, if they are in large concentrations of protein, will actually stop mating and refeed if you introduce those proteins early in mating, and so they've proved to be difficult experiments to do, but it would definitely be worthwhile to think about.